And welcome to everyone um, on a Sunday morning, a very nice Sunday morning. All right, let's start. Can you see my screen, Dr. Ranjit?
Yes, ma'am, it's visible. You can make this as a full mode.
Yeah, okay, great. I'm just
Yeah,
going to
perfectly
get
fine, ma'am.
perfectly fine. All right, let's do this. All right, so as introduced, thank you very much for the introductions. The title of my talk is Forensic DNA Testing, Challenges and Opportunities. I've been introduced, but I think the most important part in this particular slide is the email address. If there's anything that was not clear or if there are any further questions, please do not hesitate to send me an email. And I will try to respond as best as I can. All right. Forensic DNA testing in criminal investigation is a very broad field. Therefore, I would like to focus the next 45 minutes on forensic DNA testing in terms of DNA mixtures, challenges, and opportunities. So why DNA testing? Why forensic DNA testing? Well, as introduced, it is actually currently the most powerful tool for human identification. In the Philippines, I'm not very familiar with the Indian criminal justice system, but in the Philippines, over 96% of our cases being tried in court actually makes use of witness testimonies. And we know that witnesses are good in terms of criminal investigations, but many of the testimonies are somehow flawed or are not completely accurate. And therefore, we need additional means or additional evidence to provide objective confirmation of what was seen, what was heard, what was included in that testimony. So DNA provides this objective evidence. In most laboratories, DNA profiles can be generated as fast as 3 hours and in some cases even shorter. It can be, and it has been in many laboratories all over the world, been automated for large-scale analysis. And in fact, the potential of DNA databases. Dr. Ranjit, does India have a forensic DNA database? Okay. If you have, then that's good. So the potential of DNA databases in terms of efficient management of information. We know in this global in the current state of information in all over the world, it is not only just generating information, which is huge, massive, but also in being able to archive that information such that we are able to maximize first the data mining, what information we can get from a particular type of evidence. Two, how we are able to store this information efficiently such that when we need to take that information or we need to access that information, we are able to do so. So DNA as an evidence has great potential in terms of being included and DNA databases for those investigative searches such that we are able to generate investigative leads if there is no good evidence during the crime scene investigation. Since we're focusing on DNA mixtures or associated with rape cases, I tried to look for rape statistics by country to determine how much rape, the crime of rape, is affecting different countries all over the world. And in here, there's a World Population Review, and this is the website, Rape Statistics by Country 2023. And you can see here in the color is actually coded. The darkest blue, which is around Botswana, Lesotho, South Africa, are the most, have the highest rate of reported, take note, reported rape cases. There are certain countries here that may have high rape incidences, but they have not reported. So here we have, this is South Africa, Botswana, Lesotho, etc. Over here on the upper side, this is actually Sweden. And then we've got the blue, which is, this is the US. And then you've got US, this is Australia, New Zealand, etc. Here on the table, it actually states the number of rape incidents per 100,000 citizens. Figures do not take into account rape incidents that go unreported to the police. Take note that this data was actually, when I checked the raw data, these were actually not 2023, but some of them are 2004, 2009, 2010, showing that it is not easy to obtain rape statistics on a per country basis. Take note, this is India, which is in light yellow, and then the Philippines, which is in green, in light green. Again, these are numbers that need to be verified on a per country basis, because the material was only combined or compiled through reports provided by news reports or countries, etc. Now, there are many challenges in getting rape statistics, as in that particular report. Many victims of sexual violence choose not to report the crime out of shame or fear of reprisal. Some country laws are insufficient and are not regularly enforced. Victims feel that they will not get justice, and therefore silence is better. In the Philippines, we have also a huge number, or a large number, of minor victims, which means that these minor victims, the children, are incapable of going to a particular police office or agency in order to report, particularly if the perpetrator of the crime is someone known to them or related to them, such as their father, their brother, their uncle, their grandfather, etc. In most countries, fewer than 40% actually seek help. Most rapists, according to this report, escape punishment. In the US, it is estimated that only 9% of rapists are prosecuted, and only 3% actually spend time in prison. In conclusion, 97% of rapists, more or less, walk free. And therefore, as forensic scientists, this is something that this opens up a wide scope for opportunities for us to be able to use forensic science in order to improve the criminal justice system in our own countries. In the Philippines, the Philippine National Police, one of our law enforcement agencies, reported that rape cases in 2023 was actually lower than when it was 2022, it was lower than when it was in 2021. However, in both 6-month periods, theft, rape, and physical injury registered as the top 3 most prevalent cases. Remember that this period was still in the COVID pandemic, and many of the victims, particularly those abused in their own homes, have not been able to go out of their homes to report domestic abuse. The reporter also added that in the country's Peace and Order Indicator, POI, the total number of crimes dropped from 6.37 or 7,178 incidents from 112 in 2021 to 105. Again, we have to take this into a bit of a take into perspective that these numbers were actually collected during the time of a pandemic, when things were not normal or regular. I tried looking for India because I know the audience has a lot. There are a lot of members in the audience who are from India, and asked the question, what is India's rape problem? And it's reported here. I can only get a 2019 report. A number of rape cases in India in the past few months have put a spotlight on the issue of gender-based attacks in the country. Experts say deep-seated patriarchy has created a second-class status for women in India. Again, this is something that perhaps you, the forensic scientists in India, could discuss amongst yourselves and provide solutions for your own national problem. According to the latest government figures, well, it's not latest because it's 2017, registered cases of rape was 33,658. And that a woman is raped in India every 16 minutes. In fact, there were certain reports, and I pulled this one out. A lot of Indian women in the photo were asking that it is necessary now for not only for women, but for the entire Indian public to stand up for women today. Now let's focus now on sexual assault cases and how sexual assault cases are investigated and how we generate the DNA evidence that could assist in the resolution of this type of cases. Sexual assault case or rape is actually a very special type of crime, because by the very nature of sexual assault, there is close contact between assailant and victim. There is bound to have sample transferred, such as blood, semen, skin cells, sweat and saliva, and hair from the perpetrator or the person of interest, POI, to the victim and vice versa. And therefore, one clear job of a person examining, of the doctor examining the victim or those that have been able to access samples or clothing from a potential suspect, is to be able to determine or to extract DNA from either the victim's body or victim's paraphernalia and find the POI's DNA, and then vice versa, the POI's samples and bodies, etc., could also contain the victim's DNA. So therefore, the investigation of the process of transfer between one and the other is a critical piece of evidence that could be included in the investigation of the case. In some instances, and again, here I put bones and teeth, there had been a need, there was a need to identify human remains because the body of the victim has been abandoned in a crime scene. Then you ask, could there be semen? Could there be skin cells? Semen, we don't know for sure, but if there are skin cells, sweat and saliva, and hair samples, could be used to document that transfer. And if the body of the victim is no longer recognizable, then DNA amplification or generating the DNA profile of the human remain could be done using bones, or forensic odontological examination can be done, the lecture of which I've already heard in this conference. So what's the usual route? And I'll give you just a brief route in terms of how a general forensic investigation happens. There's crime scene investigation, there's a crime, and there's a crime scene investigation. Oops, all right. During the crime scene investigation, testimonies are collected from those who had witnessed the crime or had witnessed events leading to the crime. It is particularly difficult for rape cases, since rape cases are usually personal, and the people that are present in a rape case are usually those involved, either the victim or the perpetrator or the perpetrators, if this is a multiple gang rape. Difficult to collect many testimonies, but again, reconstruction of the crimes, of the sequence of events leading to the crime scene, can be done by piecing those testimonies together. Collection and preservation of physical evidence at the crime scene, at the location perhaps where the rape occurred, and particularly in a case of sexual assault, the crime scene is the victim's body. And therefore, collection has to be done by a medical doctor trained to collect samples from a rape victim using a rape kit or a sexual assault investigation kit. The samples, depending on what samples are, not only DNA, but drugs and potential urine for potential drug testing, etc., are collected and then sent to the laboratory for analysis and interpretation. And then those results are submitted to the court for the identification, capture, and trial of a suspect, hopefully would be the perpetrators, to end the particular investigation. Now, when we're talking about medical examination of a rape victim here, it is crucial that the samples are collected as soon as possible, because there is a danger of losing those biological samples that have been transferred to the body of the victim or the perpetrator. In the Philippines, collections are done within 72 hours or 3 days post-contact, so that washing, etc., to preserve the samples. In the US, there are documents that there have been recommendations to collect up to 5 days post-contact. In the Philippines, there had been certain discussions. I don't know in India, but in the Philippines, there had been certain discussions whether we will follow the US requirement of up to 5 days, because we need to consider the success rate of DNA testing. A doctor that collects sample from a victim actually would have to again go back to the locations, parts of the bodies of the victim where there had been violation. And the process of sample collection is actually, somebody raised a hand. Can you hear me? Am I supposed to answer that?
Yes, ma'am, you are very much audible.
Okay. There was somebody that raised a hand. Anyway, okay. All right. So so we need to consider the success rate of DNA testing, because if, and this is our discussion in the Philippines, if we have not shown success after 72 hours post-contact in terms of generating a good DNA profile from the samples collected after 4 days, for example, then at this stage, we do not recommend the collection beyond 72 hours for the protection of the victim. Remember, the victim has already been raped, is traumatized, and further collection, which will not produce positive results, is not something that we would like to happen. So unless you are able to make the DNA testing more sensitive, more positive after 3 days, then we do not recommend it. In the US, they recommended 5 days. Perhaps their procedures have already been validated for that period of time. Also, in the collection of samples, documentation is key. I don't know if we have lawyers in the audience, but one of the things that we need to show when we go to court is that there is an unbroken chain of custody. I have been talking to the doctors who have been collecting samples from victims, and they say that this is a particularly difficult period. Collecting samples from a traumatized victim, she's there almost naked, swabbing all different parts where the victim has to or tells you the violation that had happened, very emotional, etc. So sometimes things could be forgotten, but that should not happen, because if things are not properly documented and if samples are not transferred from the doctor to the laboratory with a proper chain of custody, with the right number of samples, as in documented, we collected 7, we collected 5, we transferred 5, we collected 3, we transferred 3, etc. And then this will open possibilities for the defense lawyer to get to question the integrity of the evidence. So therefore, this is something that we in the Philippines have been discussing with medical doctors of the Child Protection Network, those that are responsible for collecting samples from rape victims. And I guess in India, that's something that you also can do with your medical doctors. All right. So in the collection, we talked about a sexual assault investigation kit, which used to be called a rape kit. Now, the more common term is sexual assault kit or sexual assault investigation kit, which involves a lot of documentation and a lot of swabs. These swabs are used to collect samples from different parts of the victim's body: the internal genitalia, the external genitalia, the anus, if there was anal violation, oral swab if there was oral sex, or wherever the victim actually says there had been contact, and very close contact. Therefore, doctors are trained to actually listen to the narrative of the victim in order to collect those points of contact where the perpetrator's DNA is most likely to have been transferred. Once the collection, once the samples have been collected, samples are properly stored in specific tubes, envelopes, etc., properly labeled before the samples are transferred to a DNA laboratory. Okay. Okay. All right. So here, we talked about documentation is key in the Philippines, and we actually published this paper last year. So if anybody's interested to get this paper, this is open access. I can give you the citation. But the DNA Analysis Laboratory of UP, where I work, these are some of the documents that we use to properly document, properly to record what the well, we don't. Us DNA analysts, we don't. The medical doctors do. So the name of the victim, the gender, the age. We talk about gender because not only females are raped, there are also male victims that have come to the laboratories for medical examination. The most recent incident of sexual assault, which should be within 3 days. The occurrence, the address, whether it is indoor or outdoor, because then we need to understand if there are soil contamination in the samples, likely because the victim was raped on a grassy area, etc. And we need to know how to clean those externals, separate the DNA out of those externals in order for subsequent analysis. If the victim realized or felt if there was ejaculation either within or outside, or none, because that is very important, particularly for the older victims. The younger victims, and there have been victims, samples that had been collected from very, very young children, younger than 7 years old, and they do not understand this question of ejaculation. They don't understand what this is. And therefore, the medical doctors have to use their own, they have their own processes, their own protocols to be able to ask these types of questions. I personally don't know what questions are asked, but then these are provided to us. Because if we have, for example, if we've seen that the internal ejaculation was checked, then it gives us a greater hope to be able to isolate sperm cells or sperm or male DNA. Microscopy, histological examination can actually differentiate, if present, a sperm cell has the characteristic histological appearance of a head and a tail. And that could be seen. Many laboratories, but not all, in the Philippines actually do microscopic examination of samples collected from a victim. So it could give positive or negative. And here is if the victim, I don't know the psychology of a rape victim, but in many circumstances, when we've discussed this with the psychologists, they said that the victims feel very dirty after an attack, and many would actually go and wash themselves, take a bath, in order to try to remove the dirt, in quotation, the dirt that was transferred to them as a product of the rape. There's a history of sexual contact within 3 days before and after the sexual assault incident. There had been cases where a girl or a person has had consensual sex with a boyfriend or a partner or a husband, and then immediately after that, or within 3 days after that contact, she was raped. And therefore, we may see in our results not only one non-victim DNA, but 2 non-victim DNA or 2 males. It doesn't necessarily mean that she was gang raped, but that she had contact with more than one person. And all of this need to be properly documented. Relationship to victims are also checked, because we know that the DNA, we know that our DNA comes from our mother and our father, and therefore any of our relatives, particularly our nuclear family or our grandparents, etc., share the same DNA with us, not because there was contact between the victim and a grandfather, but because the grandfather shares 1/4 of his DNA with the victim. And so therefore we need to take into account the possibility that DNA was present there, not because of sexual assault, but because they were related. And so we ask the relationship to the victim. The date and time of sampling, it's all those are clear. And then here is critical in terms of when we go to court in order to testify on the samples that were tested for DNA. How many swabs were collected and placed in the tube, how many external genitalia, how many anal, etc. This has to, the numbers placed in this document has to be consistent with the number of samples that we receive in the lab. When we receive a sample in the lab, we actually receive the samples in a pouch, in a transfer pouch, not a single swabs. And therefore, we need to expect, me and my team, we inspect the pouch, we open the pouch and check before any testing is done, if the numbers shown here are actually consistent with the numbers that we see in the pouch, because there's a possibility, we don't know if there's a possibility that we could be documenting a different rape kit and a different rape. And so before anything is done, we need to make those corrective actions before DNA profiles are generated. Then we also document sample storage. Are the samples stored in a ref? That's usually recommended, refrigerator, because lower temperatures preserve DNA better and prevents further growth of fungal DNA, fungi, etc. Or the room when, say, an underwear or a clothing is dried, those are too big in a refrigerator and we store them at room temperature. And then who conducted the sampling? The medical doctor's name and when it was done, such that if we have any questions to the collection of samples, then we are able to ask those questions. In fact, in the Philippines, we don't have a DNA law that prescribes how we are going to use DNA in court. And so in the absence of a DNA law, the Philippine Supreme Court promulgated what they call a judicial rule on DNA evidence as early as 2007. This rule on DNA evidence provides guides to actually it's a rule. So it mandates the judges of all Philippine courts to follow this particular set of rules when DNA evidence is presented in court. And here in Section 7, it states, in assessing the probative value of the DNA evidence presented, the court shall consider the chain of custody, which I just described to you, including how the biological samples were collected, how they were handled, and the possibility of contamination, the method, including the procedure in analyzing the samples, and the forensic DNA laboratory, including accreditation by any reputable standard-setting institution. So all of these requirements have to be fulfilled before a certain court would accept the DNA evidence in relation to a particular rape case. Earlier, quite a number of years ago, before we really put on a lot of research in this particular area, we first tested 39 cases. These are 39 actual child cases involving females who were under 18 years old, with their guardians providing consent to the inclusion of those case samples in our research. In turn, we provided them with free DNA testing and a profile of the evidentiary samples. 39 cases, we checked if there were sperm that were seen under a microscope. We said that earlier. There were, out of the 39, 6 were positive for sperms. The handler, the medical technician, said they saw 6, in 6 cases, they saw sperms under a microscope. 21 did not see, probably because of mechanical handling. The head and the tail of the sperms were detached and could not clearly be seen under a microscope. In 12 cases, there was no report. 18 of the victims, particularly the older ones, the ones about 14 years old and above, said that they felt something warm during the contact. 5 said none. 16 could not understand the question. And this was particularly prominent with very, very young children, 12-year-olds and below. 28 said they washed themselves before they went to the clinic. In fact, not many of them went within 24 hours. So it was from 24 to about 72 hours is the more common. Day 2 and day 3 was the most common day post contact when patients or victims went for medical examinations. 8 did not wash, and 3 could not understand the question. Again, the very young ones. When we actually tested male DNA, again, these were 39 female victims, and we looked for the Y chromosome on the samples collected from the internal genitalia, and 20 of those samples were positive, and 19 were negative, and there was no non-report. So this was very clear improvement in terms of how a case could progress in court, particularly for these 20 cases. Having processed samples taken from the inside body, very intimate part of a child's body, and that showing male DNA was already very indicative of almost absolute contact with a male person. In fact, after this 39, we went up to 154. We actually published this so many years ago, 154 female child sexual assault cases following similar procedures. There were 2 to 18 years old female patients. So we had our youngest was a 2-year-old. Samples were collected within 72 hours. DNA testing was later. Remember that the collection period, 72 hours pertain only to the collection period. Samples can be stored inside the ref. So this is what we keep on telling our medical doctors. The critical part is the post-contact, because that's when samples, biological samples, and evidence could be lost. And therefore, that is the most urgent thing to do. 130 child victims knew the person of interest and provided their names. It's somebody close to them. Fathers, usually fathers, grandfathers, uncles, and brothers, and next-door neighbors. 63 cases were positive for Y DNA, again, the male DNA on the internal genitalia. 39 of this had full Y DNA profiles using a multiplex kit, which we used at that time for generating male DNA profiles. And this 39, if placed in court or filed in court, would have been very good in terms of identifying or proving or matching with a DNA profile of a potential suspect. Unfortunately, none of those cases followed up to provide a male reference profile from a suspect. This 154 cases, we talked to the agency involved, and those individuals, those children, did not file a case. And I guess, again, this is beyond DNA testing, but the problem is more complex than just reporting. To talk a little bit about the technical part, when we do DNA testing, we actually do short tandem repeats, wherein when we do DNA testing, we actually extract DNA from the sample collected from the victim, and we amplify or increase the amount of DNA using a polymerase chain reaction. It's a standard molecular procedure targeting what we call short tandem repeats, or STRs. These are regions of our DNA, of our human DNA, that is characterized by short segments of nucleic bases which are repeated, and that's why we call them short tandem repeats. What we do in the laboratory, in the first technology, is only to count the number of times these STRs are repeated per chromosome. So here in this particular example that I took from John, I think this is John Butler, this figure, there are 4 repeats, 1, 2, 3, 4. There are 5 repeats, 1, 2, 3, 4, 5. There are 6 repeats, etc. So then when we try to identify, or we generate the DNA profiles of the persons that own this DNA, the differences in the number of repeats per chromosome, or in a bigger sense, one can say length variation, because the more number of repeats would have a longer length, then that would be the DNA profile of that particular person for that particular gene or DNA marker. Again, we'll see this a bit later on. But remember, this is what we're doing. We're simply counting the number of repeats in a particular site of the DNA of the victim or the perpetrator. And then once we do this, just like a simple play of cards, when you do cards, then when we have the number of repeats, we know what the profile is of one person, then we check the evidence. Does the reference profile of the person match that of the evidence? Is this the same card? Is this the same Jack of Hearts? Are they exactly the same? Or are they not exactly the same? No, one is a jack of club and not a jack of hearts, and therefore there's a difference. Okay, we published this last year, and again, this paper is open access. If anybody's interested to get this, you can actually get this online. Forensic Science International Synergy. The title is Unintegrated System for Forensic DNA Testing of Sexual Assault in the Philippines, published by Rodriguez and co-authors. There are three main sections of the procedures. DNA testing of DNA mixtures is not easy. It's one of the more complex types of analysis. So what we did is in this particular paper is we grouped the steps into particular sections for easier understanding. First is the sample collection, storage, and characterization. And I will go through these steps one by one. DNA extraction, amplification, and fragment analysis. So the collection happens in the hospital, in the clinic with a medical doctor. They store it and then afterwards submit it to us. This section on DNA extraction, amplification, and fragment analysis happens inside the laboratory. So we do all the work inside the lab, extraction and do the lab stuff. This next section here, which is the DNA data and evidence interpretation, happens outside of the laboratory, but in your using your computer. And again, we'll go through this step by step. All right, let's start with this first one. Sample collection, storage, and characterization. We basically touched on this already. There's a sexual assault investigation kit, the rape kit. The specimens were collected by the medical doctors. Then our first question here in this flow chart. With feces, yes or no? Then you ask, why do you talk about feces? Well, if the swab comes from the anus, particularly in children, then there are certain swabs that are actually have a high proportion or a considerable amount of contaminating feces. If there is feces and it's a good amount, then we need to store that particular swab at 4 degrees and actually process that first amongst all others. Why? Because when you have, when the sample has feces, it has a lot of microbial and potentially fungal contamination. Microbes and fungi actually replicate and have DNA and could prevent us from being able to generate the human DNA from the sample through non-competitive inhibition. If the samples do not have trace amounts of feces, then we can store the samples at RT. Again, we may, or actually we choose to store them at 4 degrees, but there are certain laboratories that do not have that options, and therefore we tell them, yes, store them at RT if there is no other option. Then we check for the type of material. If it's a vaginal or anal swab, then that goes into this SG test. Is a 𝐬𝐤𝐢𝐭 test. I'll tell you about it in a while. So here, let me just go back. This is a swab from internal vagina. This is an internal vaginal swab. You can see it's more white, yellowish type. This is a fecal swab. This is an anal swab which has traces of feces. So actually a good proportion of feces. This is the type of samples that I will tag as red, extract immediately. All this description here is actually talks about what we call a presumptive test. A presumptive test is a test that when it goes positive could possibly indicate that there is something worth testing. One example of that is the alternative light source, such as this one, alternative light source with orange goggle. And here you can see it fluoresces. Here, this is underwear with that has sperm in the crotch region. Under the alternative light source and the orange goggle, you can see locations where the semen is, and that's where we will cut samples for DNA extraction. Alkaline phosphatase test also is a presumptive test commonly used by police agencies, etc. And there are those which are classified as confirmatory tests. And I talked about 𝐬𝐤𝐢𝐭. 𝐬𝐤𝐢𝐭 is a semen-specific protein. This is found in protein that when you detect or one detects 𝐬𝐤𝐢𝐭 in your sample, then that is already indicative that there is semen present. This is the type of 𝐬𝐤𝐭 test, as you can see here below. This is like a pregnancy test. So this is your positive double band. This is negative, only the control band is there. But these are your other samples. So this sample is negative. This sample potentially could be, I could see a little bit there. This is definitely positive, and this is definitely positive for semen. So back to the flow chart. If we go and check the alternative light source, the condom or stain will have to check if it's blood stained. If it's blood stained, then we do not bother with an alternative light source because blood actually interferes, the color interferes, and we would definitely go for an alkaline phosphatase test. For the internal and external swabs, we will definitely go for some 𝐬𝐤𝐭 tests in order to determine if semen is present in those samples. And then we proceed to the DNA tests, the laboratory samples. So in the laboratory stage, DNA is extracted from the samples. Again, we are talking about different samples: internal vaginal swabs, anal swabs, oral swabs, etc. And processing. Now, one thing that our laboratory, my lab at the University of the Philippines, have actually opted not to do a differential organic extraction step. I'll briefly describe it here, and I'll tell you why we don't do it, but many other labs do it. So this is something that you need to determine what works for you. A differential organic extraction procedure actually targets to have two fractions of DNA. A female fraction and a male fraction. What happens here is that swabs are put inside a tube and then incubated for 1 hour or 37, 1 hour for, at 56 or 37 degrees for 2 hours or overnight. What happens here is that using this step, the sperm actually, if it is intact, will pellet out to the bottom of the tube. And the epithelial cells that would come from the female, these are her skin cells, would actually separate out into the solution. When you spin this in very high speed, centrifuge, one could take the fluid out of the tube, that's your female fraction, and then the remaining part of the tube with the pellet remaining at the bottom will then be further lysed, will be taken and dried, and then the sperm cells will be lysed to get the male fraction. Now, one thing, and you end up with two fractions, the female fraction, and you can check for the DNA profile of the patient or the victim, and the male fraction, which you can check for and compare with the reference profile from a suspect or person of interest. The problem that we've had in our experience with the differential organic extraction is that we lose a lot of DNA using this procedure, and therefore end up with very trace amounts of DNA that does not provide us with very good DNA profiles. So in our lab so far, what we've done is we've actually done a full extraction without separating the DNA male and female using the 𝐸𝐴MP DNA mini kit. And so one extract, total DNA. So once we've done this, we go back to this particular part of the flow chart, and then ask, we quantitate the DNA, and in the quantitation using real-time PCR, this is a standard procedure. Using this procedure, we can actually quantitate the amount of male DNA and then the amount of total DNA. Then we ask the question, is there an excessive amount of female DNA in the solution? If the answer is no, we then proceed to a full-blown analysis targeting the body chromosomes or autosomal STRs and the male-specific, because the DNA of the victim will not interfere as much with the DNA profiling because they're almost about similar in quantities. However, if there is so much excessive female component, then we target the Y chromosome or the male specific. And then we run this on the machine. Showing you the readouts of what we generate in the machines. This is we call them electropherogram. This graph is an electropherogram. The proportion is 0.25 as proportion 100 male female. So there's a lot of female DNA compared to the male DNA. So what we did here, as I've said earlier, is that we targeted the male DNA profile. Even if the sample has excessive amounts of female DNA, it doesn't matter. Why? Because the reaction only targets the Y chromosome or the male specific, and the excess female will not amplify, it will not provide a result. So using that parameter, we were able to generate a Y DNA profile or a Y DNA haplotype of the male DNA found in the sample of the female patient. And you can see here is very clear that we have signals for those DNA markers in the sample. And that is the Y genotype. Because, and this is for those interested, because males have XY as the sex chromosome, females has XX, males only have one copy of the Y chromosome, and therefore in most DNA markers, you will only get one peak because there is one copy. Pero what happens if you have two peaks? Well, then if you have two peaks here, then you know that you have two males in that sample, and you need to review the interview, the information provided by the victim. Did she have consensual sex before the rape? Or was there any possibility she was drunk or drugged such that she didn't know there was an additional person that raped her? This particular marker, this is DYS 385. There are two peaks here. This particular marker we know has two copies, unlike all the other DNA markers on the Y chromosome. This particular marker has two copies, and so we are not alarmed. This is a DNA profile of one male individual. Then we can go, so that was the Y chromosome. A reference profile, then we can generate a reference profile of a male DNA profile. For example, if a suspect provides a sample willingly or as following a court order, not a mixture, but a sample, a buccal swab or a blood sample, this is what a DNA profile of a reference material looks like. XY, he's male, and then all the different amplifications throughout his DNA. The two peaks means that he has different number of repeats on that particular marker. One peak means that he has the same number of markers or STR in that marker, and he's homologous for that particular marker. This is the DNA profile of this particular sample summarized on a table. And this is what we will use to compare with the DNA profile of a two-person mixture. So we know that the maximum number of copies in our autosomal DNA is 2 per person, except for some cases of abnormalities. But a normal person would only have maximum of 2 copies per DNA marker. And in this particular two-person DNA mixture, you're seeing more than 2 peaks. This is 1, 2, 3 peaks. This is 1, 2, 3 peaks. 1, 2, 3 peaks. 1, 2, 3 peaks, which actually tells us, hey, look, you have more than 1 person there. And so the two-person DNA mixture in a rape case would normally have the victim DNA, which is female. That's why you have a very high peak of X here and a very small Y, because a female would have XX and then 1X to the Y of the male. And then a person of interest which is a male. As you can see, there's a Y peak there. And this is what we term as an evidence sample. This is a sample that we need to analyze in order to support any claims of rape, the allegations of rape. Okay. Now we go to the third part. So we've extracted the samples, we've generated DNA profiles inside the lab, we extracted the DNA and then generated the DNA profiles. Now we need to go to the computer and analyze what does this mean and interpret the data. Now this is something that's still ongoing in our laboratory in the Philippines because we have not really fully understand the complexity of the analysis. There's somebody that raised their hand. Am I supposed to answer that?
No, it's I think by mistake.
Okay. So right. So this is data interpretation. So we've got, again, autosomal. This is your normal body, the body chromosomes, the 22 pairs of chromosome of each individual, or the Y chromosome, which is only for males. If we're doing, and let's just focus on the male chromosomal DNA first. If we analyze the Y chromosome, the male chromosome, and we see that there's a single source, the recommendation by Dr. Sasha Wiluit and Lutz Rower of the YHRD group is to conduct a single source, a discrete Laplace model, Y haplotype frequency using a disclap mix R package. Honestly, we have not been able to, in my lab, we have not been able to fully finish this analysis. And in fact, if you go to the YHRD, we are being helped by Michael Anderson to be able to do this types of interpretation. So this is ongoing. If there is more than one source of Y and there's a mixture, there's another recommendation, again by the same group, to use YHRD mixture analysis tool, which is available on their webpage. And I put here their webpage. For those who are more computational savvy, go and have a look at the YHRD mixture analysis tool. Then if we analyze the autosomal STR, the 22 pairs that we have, then we actually will recommend the use of a continuous model of interpretation, and to be able to differentiate, and I'll show you again. When you have a mixture and you're analyzing here, when you see a graph like this, one needs to be able to differentiate which DNA profile is that of the victim and which DNA profile is that of non-victim. Why do you need to do that? We need to do that because when we have the DNA profile of the person of interest from the mixture, that is the DNA profile that we will compare with the reference profile of a suspect in order to know if the reference profile of a suspect matches that of the evidence. In order to do that, we need to do this continuous model analysis. And that's also happening in my lab at the moment. In the Philippines, we have not really gone full on in terms of computational analysis of DNA mixtures. In the past, based on our work, we have simply been citing that there is DNA present, male DNA present in the profile. We're checking if it matches that of the suspect, but then we have not been able to generate likelihood ratios or statistical analysis. This statistical analysis is important because that gives us us and the judge an estimation of the likely weight of the match, as answering the question, is that match a real match? Really, is the match because he was the source of the sample, or was that a random match, a match that happened because the DNA profile in the mixture is so common in the Philippines such that matching the two DNA profiles does not really tell us much? So in order to do this, we are currently, and we have a research project studying the features of two probabilistic genotyping software. 𝐕𝐢𝐬 is a freeware, and an MASTR mix is actually something that we purchased. So this is you can see from the 𝐕𝐢𝐬, you can actually access this online. It's a CRAN-R program. The input file is from a gene mapper IDX, which comes from our computer in the lab. The interface is not easy. You really need to do a lot of programming and R in order to be able to proceed with this particular software. There's no job queuing, and the required analysis input and time is very heavy. It's time consuming. Downstream is step-by-step user response, and also considers degradation, the possibility that the samples have actually lost signals because you have stored it for too long, or that there was a nuclease, an enzyme that was eating up the DNA during storage, or that there were stutters, i.e. these are just random peaks that happen in a mixture and therefore should not be considered. In the MASTR software, the code, of course, they don't tell us, it's commercial. It can be done on Windows. It needs formatting, but we like it because it is user-friendly. It's a graphical user interface. we get a lot more visual representations of what we're doing and are able to understand the results better. We need, though, to job queue a lot of our analysis, but the input of the analyst is minimal. And we can do all this downstream. Now, why are we recommending a fully continuous probability models? There are two other models, a binary. I think some of you, those that have
Ma'am, we are not getting you.
maximize the use of all most or a lot of laboratories now are recommending and are developing and pushing the use of the fully continuous probability model because it allows us to use the LR approach, the likelihood ratio approach. This is a statistical estimate to discriminate between true and false propositions with the amount of correct information provided. This is achieved by maximizing DNA recovery of the semen contributor to the mixed sample and in employing the method which considers much of the available information from the graph. When we were using the other models, such as the binary and the semi-continuous, there were certain outputs of the graph from the laboratory that we had to ignore. Don't do that. Don't include that. It just makes it more complicated, etc. And therefore, it does not maximize the information that one can extract from the graphical output. The fully continuous probability model actually makes use of all the peaks and information in those graphs in order to model and train. It has a training set. It trains itself to be able to distinguish between a real peak and a false peak. And therefore, that is what is being recommended now. In fact, last year, in the Asian Forensic Sciences Network, I don't know if people from India are going to the next AFSN meeting, which will be held in Kuala Lumpur early next month. I think it's September 11 to 15. My team at the University of the Philippines lab already reported the analysis of a 19-year-old sexual assault case of a minor, arbitrarily called 44, using Euroformics, a software, probabilistic genotyping software. In the poster that I cut, it's a much longer poster, but we actually compare the binary method, which I just talked about, the semi-continuous method, and the fully continuous method. And this 19-year-old case involving a 12-year-old female patient that was collected by the Doctors of the Child Protection Network had a full profile 19 years after contact, after submission. So the child by this stage would be about 31, 32 years old. Unfortunately, as we have said earlier, unfortunately, this particular case was not filed in a criminal court, and whoever did the crime did not pay for the crime. But anyway, the poster is in that particular meeting. Going towards the end of this lecture, for those, the ones that are in the laboratories, I highly recommend going through some of the documents that are critical in terms of validations of your procedures for DNA mixture analysis. I could not go through all of these in this presentation, but this is an ASB standard 2016 standards for validation studies of DNA mixtures. One needs to test your own computers, your own equipments for thresholds, analytical thresholds, stutter thresholds, etc., to determine which section of the output you should include in your analysis. The group of NIST, the National Institute of Standards and Technology, headed by Dr. John Butler, has also produced a foundation review of a good proportion of literature on DNA mixture interpretation. Again, highly recommended. You can download this on the internet. And for those ones that have not generated yet DNA mixtures, but would like to try this particular softwares, there is a data set, DNA Mix 2021. This is a data article with supplementaries that you can download from DNA in brief, and you can use the actual data to be able to understand the softwares that I've just mentioned in the last 2 minutes. Another technology that has come up, although we don't have time to be able to discuss this, is what we call massively parallel sequencing. Massively parallel sequencing is another technology that allows us to understand or to characterize the DNA profiles of different people, such that we can differentiate the DNA profile of the victim and the DNA profile of the person of interest. But this massively parallel sequencing technology claims, and it is actually, that it has a higher capacity to discriminate. Remember, in the previous technology that I was talking to you about, we were talking about length variation. Depending on the number of repeats, one can say that two persons are different. But what happens if two persons actually have the same number of repeats? Then you could not differentiate them in a particular marker. What massive parallel sequencing is, and you can pull out this particular paper and other papers, there are a lot of MPS information now, is that massively parallel sequencing not only characterizes the length of the DNA, but also the sequence, the internal sequences of those DNA markers, of those repeat units. And there is what you call intra-allelic sequence variations, such that two persons can have the same number of repeats, but the sequences of the repeats inside them are not the same. And therefore, this is provides you with a higher capacity to discriminate between persons. Again, that could be something for next year's conference, Dr. Ranjit. So now let's go in closing towards the end. What are the challenges in investigating rape cases? And in this particular slide, I talk about rape cases involving minors. There is delayed reporting, if at all. And as I've said, in those 150 odd cases, nobody filed a case because many of those persons that they identified in the narrative to the social worker is somebody related to them. And therefore, there is definitely an area of fear, of shame, and also probably that they were threatened not to report the abuse. And likely those abuse is not a single occasion, but a repeated abuse over a period of time. The emotional burden, we talked about shame, we talked about fear of reprisal, dependence of an adult to seek assistance. Many of those kids, particularly the younger ones, do not even have the money to take a jeep or a bus to go to a clinic because they are dependent on their fathers to provide the money, and therefore they cannot. Others are dependent on their mothers, and we've heard stories that mothers would also tell the kids not to report because if their fathers are taken to jail, who will feed the rest of the family? So again, economic vulnerability. In some of the kids, particularly the 2-year-old, the 3-year-olds, they don't know what happened. They don't know who. They don't know anything. They just say it hurts. And then also the challenge, because when body of a victim is smaller, there's less DNA that could be collected, less sample that could be collected. Okay, so towards the end, one challenge, if we do not maximize the use of forensic evidence, including DNA evidence, which has the power to identify sources of samples, be able to link persons to crime scenes or to victims, is that in the Philippines, the occupancy level of Philippine prisons was more than 400%. And in 2020, in the World Prison Brief, we were the second highest level below the Republic of Congo. In fact, these are photos of some of our jails that have already been shown in the newspapers, and we know this could actually increase. In this particular date, it actually says 314. In the later one is 400%, etc. This is not a park. This is actually inside Quezon City Jail, where the jails are insufficient to house the prisoners already. And so this is how they would stay awaiting their trial. Many of those cannot afford bail or are actually being held on non-bailable offense, and therefore they have to wait. Now, one challenge with our trials is that a trial of a case, this is estimated by our Supreme Court, a trial of a case actually averages from 4 to 7 years before a decision is obtained. And therefore, we are actually jailing people without a proper sentence in case they are not the right persons. And so I'd like to end with some of the material that we've actually been telling over and over again to our policymakers, to our agencies, that DNA testing of sexual assaults surmounting challenges in the Philippine setting. I don't know if India has the same challenges, but here we're talking about DNA analysis provides the most powerful tool for human identification and has been in the Philippines since the late 1990s. Unfortunately, DNA testing has not been used routinely. The failure to properly collect, to store and analyze biological samples had prolonged the suffering of a victim and her family. Moreover, other crimes could have been prevented had the real perpetrator been identified and incarcerated. We also push and insist that forensic DNA technology is a powerful tool for judicial reform. If we can do a DNA profile in 3 hours, if we can do an analysis of a rape case in a week, then those persons that have been wrongfully identified can be set free. And in fact, this is what we said. Recent advances in forensic DNA testing are now paving the way for reforming the manner by which cases are resolved in courts of law, through the way suspected offenders are apprehended during a criminal investigation. In my more than 20 years in the profession, we have actually participated in cases where the persons that have been accused of rape have not been the persons who raped the victim. In fact, I think our running count now is about 15 acquittals, 15, in the cases that we've already participated in, conducted. And this ends my talk. Thank you and stay safe.